whole fiber on the final, and then sometimes he has none of them. Sort of varies. But for sure, questions that are there are area specific. You know, they are areas that you'll have to think about. So you need to be sure you take a look at those and go over them. And remember that 100 points on this next exam is this material. And 100 points is the last section, quiz 4. Okay, do we have questions? Okay. Okay, so we've been talking about proteins, protein folding, and protein kernels. And the first thing I told you is that cells try very hard to fold proteins with a lot of energy into making them. They want to fold them correctly. And if they don't fold, if they in the endoplasmic reticulum form, there's a buildup of unfolded proteins. It's called the unfolded protein. We went over this before. If there's unfolding protein, it recruits a protein that normally keeps the sensor, a membrane-based binding protein, a monomeric singular. Protein is called BIP, binding protein, which is one of the folding proteins. There's a lot of misfolding protein. It recruits BIP from here, right? There's the BIP. And it leaves this sensor there without anything bound to the luminal side, the luminal side of the sensor. Nothing bound, it can dimerize, and the dimerize itself phosphorylates itself, and that phosphorylation activates it and turns on a nuclease activity. And that nuclease activity processes pre-messenger RNA, an unprocessed messenger RNA in the cytosol. And when it processes it, it becomes an active message, and it's translated into a transcription factor. A transcription factor binds to a subset of promoters in the nucleus, and induces transcription of that subset of genes. The messenger RNA, or the message that it transcribes, goes out into the cytosol and is translated into proteins. The proteins that are translated have signal peptides, and they're directed into the endoplasmic reticulum. The genes that this transcription factor activates are all genes that code for chaperone proteins. So this response increases the number of chaperone proteins in the ER, so they can more effectively fold proteins that are misfolded. You notice that these steps are numbered 1, 2, 3, 4, 5, 6. And I made a table here that shows you 1, 2, 3, 4, 5, 6 and tells you exactly what I just said. You can have questions and just sort of outline all of that. Okay, so that's the unfolded protein response. So that when proteins just can't be folded, but they have to be turned over. And they're turned over by targeting them either to lysosomes or more usually in the cytosol of the cells, a structure that's called proteasome. And a proteasome is a structure which digests proteins. And it digests several kinds of proteins. One that we just talked about, unfolded proteins, proteins that are unfolded. Secondly, damaged proteins, proteins that break or tear or whatever are misused. Thirdly, it actually has a role in digesting or turning over proteins that are needed at specific times during the cell cycle. We talked about that. Cyclin proteins, which build up during the cell cycle when the cell is dividing through the division cycle, and then allow that cell to enter the division. As soon as they do that, they're degraded by this turnover system in a proteasome. So normal proteins or functional proteins that have a cycle during the cell cycle are turned over like this. And then finally, proteins that are aggregates. So many times, with protein, we said that we talked a little bit about protein folding, and Adam's probably talked a little more. But protein folding, when proteins fold into a native form, that native form, which is the functional form, also is a form at which all of the hydrophobic residues are buried, all the charged residues are on the outside, or many of the sulfhydryls have formed disulfide bonds. And the bonding energy is minimal. Right? So they minimize the amount of energy to hold that protein together. That's the native form. If any of those things fail, as we talked about before, then proteins aggregate. And aggregates are very bad for cells. They build up in cells, and they essentially cause cells to dysfunction. So those have to be turned over also. They're all turned over by this ubiquitin pathway. In the ubiquitin pathway, there are 3 proteins. The first one is called E1, is an activating protein. What it does is it activates the ubiquitin. It takes ubiquitin, right, and which is a protein, and the carboxy terminal, and it activates one of those carboxy oxygens by adenylating it. It adds an AMP to it, adenosine monophosphate to it, and that activates this bond right in here so that the biochemistry can occur. Once this is activated, it's actually attached to that E1 protein. So you have E1 with this thiol ester, that high energy thiol ester attached to ubiquitin. And that's all that this E1 protein does, is forms this high energy link between ubiquitin and the cell. Then passes it over to the carrier protein, which is E2. E2, right, so E1 just passes this ubiquitin thiol ester over to E2 to create this ubiquitin thiol ester. E2 is your carrier. E2 and E3 then act together. E3 is actually an enzyme that's called a ligase, a protein ligase, or a ubiquitin ligase. And what it does is it takes that ubiquitin off of the E2 carrier, and it attaches to a protein, to a targeted protein. And we talked about that attachment before. Ubiquitin is a protein that has 76 amino acids in it. And at the carboxy terminal end, when it's activated, it's carboxy terminal end. Ubiquitin is attached by the carboxy terminal end to a lysine group in the offending protein, whether it's, aggregate or whatever. The amine in the lysine is attached to this ubiquitin there. Ubiquitin itself has a single lysine in it, so another ubiquitin can attach to the lysine in ubiquitin, and it generates a polyubiquitin molecule, and that serves as a flag or a tag or a signal that this protein is to be degraded. Okay, everybody still got that? Do we have a question? This is a cartoon. Oh, no. So first let me ask this. How do we know which proteins here are tagged with ubiquitin? And there are several ways. Here are just a couple of them. Some proteins are not tagged. Those proteins that have a methionine on the amino terminal end, right, are all mean that the amino terminal end is intact because it has a methionine on it. It's newly synthesized. There are other proteins that have serine, alanine, threonine, and so forth on here, right, on the amino terminal end. All of these guys are also excluded from ubiquitination. But if any of the amino acids here listed, if any charged amino acid or any of these aromatic amino acids occurs on the amino terminal end of a protein, that means that that protein has been broken or degraded. Because normal native proteins never have those amino acids on the end. So there are several of these E3 ligases, different ones, that recognize the charged amino acids or aromatic amino acids. And when they do, that's a signal for them to add ubiquitin to the lysine in these guys there and target them for degradation. The acidic amino acids, you notice these are basic and acidic. The acidic amino acids first have to be targeted by another ligase and by an enzyme which actually puts a arginine residue onto the end of these. So if there's an aspar glue on the amino terminal end of a protein or a peptide, that protein's been damaged, it's non-functional. What first happens is there's an arginine transferase molecule, arginine protein transferase that transfers this arginine off of arginine tRNA. So a charged arginine tRNA molecule, it transfers that arginine from there, right, tRNA, to the protein. So it's a tRNA protein transferase. And it just puts the arginine on the end. Once that arginine's on the end, then once that arginine's on the end like that, then it's targeted for ubiquitination and it turns over. So these E3 ligases, there are a family of E3 ligases. There are about 20 or so that we know of. And all of them recognize something unusual about a protein. There's some that recognize exposed hydrophobic regions. There's some that recognize other forms of aggregation and so forth. But all of the E3 ligases are essentially those that monitor protein integrity, monitor to ensure the protein is native state, that it's folded correctly, and If it's not, then they direct the ubiquitination of it. Question. B1E3 ligase. Once these things are targeted, you move into this structure that we call the proteasome. And this whole proteasome structure is 26S in size. It's called a 26S structure. And it's about somewhere around almost 1 million Daltons. The 4 rings in the middle are known as the 20S proteasome. So the whole thing is 26, inside is 20. Remember, S refers to sedimentation coefficient. So it's how quickly they sediment through some sort of a density gradient. It doesn't